In Chapter 7, we're going to be looking closer at metabolism and the role of enzymes. So what is metabolism? I'm going to pause. You can pause the video, take a moment to think about what you would answer in class about what metabolism is. Okay, so the answer I usually get is students usually say, well, metabolism is going to be the way that we lose weight or how we deal with our food. And so that's not exactly incorrect, but it's not completely correct either. That's about half of it. Metabolism is the sum total of all the reactions. So what most people are talking about when they think of metabolism is catabolic reactions, where you're breaking something down that you've eaten and you are making it into smaller molecules and getting energy from those processes. Okay, so that's half of it. The other half is going to be the anabolic ones, where we're building up or making larger molecules. Okay, and so most of all reactions are going to fall into one of these two categories, anabolic or catabolic. Anabolic or anabolism can also be called biosynthesis. Synthesize means to make. So this is the building up of larger molecules from smaller parts, and those smaller parts we often call monomers. So we're making macromolecules by forming bonds between smaller pieces. And you can see this in this first picture down here. I've got 4 individual pieces. I'm adding energy. And then over here, I have got a 4-piece, but it's all bonded together. Okay, so anabolic, I want you to think assembly. And I usually tell students, especially students that have had kids, on Christmas Eve and you buy a toy, which nowadays a lot of them come put together, but back when I was growing up, a lot of times you had to put everything together. And so parents would have to stay up half the night and assemble these and put them all together. And what does it take to assemble something? Energy, right? It takes energy to build something. So you're putting together something and it's going to take energy, okay? Catabolic is basically just going to be the exact opposite of that. You are breaking the bonds of a larger molecules. You're taking this one larger molecule, and you can see this is the exact opposite, and you are going to break it down into smaller pieces. And if you'll look, the energy is on the product side. So let me say this first. Anything on the left of this arrow is going to be a reactant. So you're putting in these 4 molecules, you're putting in energy, and you're going to get your product over here. And so sometimes students tend to think that a whole compound is a product, but it's really left or right hand side of the arrow, and that's all there is to it. This is the left hand side of the arrow, so this is our reactant, and we're going to break it down into these 4 smaller molecules and energy. So those are our products. All right. And so catabolic, you're breaking things apart and you're releasing energy. And so kind of the analogy I use here, not the best thing to do, but a lot of people, when they get angry, what do? They like to tear things apart, okay? And it releases energy, right? So don't go try this at home. But so catabolic, think catastrophe, things are falling apart. It's energy is being released, okay? So remember, energy is stored in those bonds. And this is kind of only partially true chemically, but it's fine. It's good for us. And you break a bond, you release the energy that's stored in that bond. Okay. And so that's catabolic and anabolic. Let's see. I think that's all I wanted to say here. The cell is going to create and spend energy by transferring electrons from an external source to an internal source via a carrier shuttle molecule that is going to drop those electrons off at a series of proteins to create energy. Metabolism, you can also say, assembles smaller molecules into larger ones needed for the cells using ATP to form those bonds. It also degrades or breaks down macromolecules into smaller ones, releasing energy, and then we create and spend that energy in the form of ATP. So here you can see this is a nutrient from outside or from an internal pathway. So outside we would say glucose. I've gotten this maybe perhaps by eating something. And then I'm going to have some catabolic reactions here. Catabolic catastrophe, I'm breaking something down. Why am I breaking it down? Because I need energy, okay? And these pathways are going to be things like glycolysis, which you should have learned about in anatomy, the Krebs cycle, respiratory chain, which we haven't talked a lot about, fermentation, okay, respiratory chain, think electron transport chain, okay, so that yields energy. We have some precursor molecules. Then we've broken this down into things like pyruvate, acetyl-CoA. You should recognize that glucose going to pyruvate and acetyl-CoA, and glyceraldehyde 3P. That's our G3P. We did go over that in the steps of glycolysis. And then we're going to have our anabolic reactions that need energy. So we've broken these things down into pieces, but we also need to make things. We've got to make amino acids. We need to make proteins from those amino acids, right? That's showing this step over here. We need to make sugars and nucleotides. ATP, remember, is actually a nucleotide. DNA and RNA. Fatty acids, okay? So those are our smaller monomers, our building blocks. Then we're going to take those building blocks together, put them together, still anabolic, and make those larger macromolecules. That's your proteins, peptidoglycan, the RNA, and DNA. And see, so your nucleotides are what are the smaller pieces of your DNA and RNA, okay, and more complex lipids, more fats, all right? And so this over here is talking about spontaneity, and what that just means is that those are some reactions occur on their own, and spontaneity usually means without the input of energy. So I'm not quite sure what they're getting at with this one particular picture in this textbook, but maybe they mean the literal, or sorry, the everyday connotation of the word spontaneous and not the chemical definition. They mean they just occur. So some assembly, it's talking about things occur on their own without us having to initiate them. We could not complete all of the required chemical reactions needed to sustain life without a special class of molecules we call enzymes. Okay, so enzymes are catalysts that increase the rate of a reaction without being used up, so they're recyclable. They're not part of the reaction themselves. So this is important because they wouldn't be much help if they were used up, if we had to constantly make them. So if we used them up in the reaction, if they were not recyclable, they wouldn't be saving us energy. And nature is all about conserving energy. It wants to spend the least amount of energy possible, okay? So enzymes can act basically kind of as the physical site where the substrates are going to actually kind of be positioned. You can see that in this little picture down here, that are going to form the active site for a reaction to occur. So of course, then they're going to be larger in size than the substrate. They have this unique active site that is specific to the substrate, and the substrate is just what we're wanting to work on. It can be one thing, or if we're doing a catabolic reaction, we're breaking something down, which is what this particular picture is illustrating. Or, excuse me. Or it can be several things if we're doing an anabolic reaction, trying to build them up. Okay, but the substrate is just, so the substrate is just whatever we're wanting to work on. It can be that anabolic. This may have recorded twice, so sorry if it did. Anabolic reaction, we have several things coming in to put together into one thing, okay? And so that active site is going to be specific to, it's going to fit, it's going to be almost like a puzzle piece fit for those particular substrates, and it participates directly in changing, helping to change the reaction, okay? This is a good little list here of some of the different characteristics of enzymes. We're going to go over most of these. This is really important. I'm not going to just read straight to you. There's no point in that. But I would be sure that you understand every part of this. And if there's something on here that I don't explain for some reason, send me an email. I will absolutely explain it to you, okay? We've already talked about them being a catalyst. They speed up the rate of reaction. We're going to get into that in more detail, okay? Enzymes are mostly protein, but there are some that are made of RNA. Some are very simple, which can be just a protein component, but many of them are conjugated, meaning they have an additional non-protein component, all right? So a conjugated enzyme is what we would call a holoenzyme, and that's probably how you've heard me refer to it in the past if you've taken my class. So holoenzyme here, think this is the whole thing together, okay? And so we're going to look at the different pieces here, all right? Holoenzyme is the whole thing, okay? This is a protein component plus one or more cofactor or coenzyme, all right? And so we can have a metallic cofactor, which is going to be a metallic ion, which is one of those essential essential nutrients that we need in a very, very small amount. A trace element, we need it in a very, very small amount, but it is very important, and this is why it is so important. These coenzymes are going to be organic molecules, and so we're going to have either an organic molecule or a metallic ion, and just the source of that is where we get the different name of cofactor or coenzyme. So if it's a metal ion, it's a cofactor. If it is an organic element, an organic compound, then it's going to be a coenzyme. That's the difference there. All right. So they actually help to form the specific shape of the active site or help the substrate to bind. Okay, so the enzyme cannot do their job without these little helper molecules. All right, so apoenzyme is going to be the protein component. That's going to be this big purple blobby part here, okay? And then the cofactor or the coenzyme then is that little helper molecule that usually helps to form the shape of the active site. And I like to say the active site is where the action happens. That is where that facilitated chemical reaction is going to occur. So for a reaction to occur, a temporary binding or union of the enzyme and the substrate must occur. So, of course, we give that a different name because that's how science is. We got to make it more complicated than it needs to be, okay? So remember this, the fit is very specific, okay? So this is one substrate. You can see it would fit perfect like a lock and key, okay? And this one here, this substrate would not fit, so they have to match. Now, we say lock and key, and our textbook only keeps it that way, and that's actually kind of the old explanation. We now understand it's more induced fit, and it's more like if you're shaking hands and when you shake somebody's hand, what do? You clasp, and they fit together much more perfectly. That's really what an enzyme substrate reaction there. I don't hesitate to use the word reaction, but their union is more like the clasping of hands together or a hand fitted in a glove. Okay, it's going to fit around it even more correctly as it gets closer together. Okay, so when they do that and they bond, then we're going to have what we call this enzyme substrate complex. So now that it's bonded together, enzyme substrate complex, it just means that the substrate has fitted in like that hand in the glove to the enzyme. All right, a bond has formed, and it's going to be a very weak bond that's easily reversible because we need it to be so. So once the enzyme substrate complex has formed, then the designated reaction is going to occur on the substrate, whether that's putting two together or breaking apart, and this one is breaking apart. So which type of reaction is that? Is that going to be catabolic or anabolic? All right, well, we're breaking it down. So catabolic catastrophe, right? We're breaking it down. So this is a catabolic reaction here, okay? Just checking. All right, so let's see. That designated reaction occurs. We break it down with the aid of that cofactor, and then the product is going to be formed and released, okay? And so if you'll notice the enzyme here, the enzyme at the beginning and at the end, it's unchanged. So remember that it is a helper here. It participates by facilitating the reaction, but does not participate in the reaction. And it's unchanged, and it is recyclable, okay? It is not changed in the reaction. Enzymes also can often catalyze directions in both ways. And so you could put these two pieces in here and go back and reform it into one, because it's still going to fit. It's still the same substrates, basically. But we usually look at them in one direction just to kind of simplify things, although sometimes they are reversible. Just kind of FYI there, okay? So at the end, the enzyme is free to interact with another substrate. Microorganisms, the reason they need those trace elements again is because of their role as a cofactor or enzyme. This is why those trace elements are so important: iron, copper, magnesium, zinc, cobalt, selenium. All right. When you have things like a coronavirus, which is one of the causes for common cold, even though the current strain is kind of worse than a common cold, you can actually take zinc, and it's proven that zinc reduces the lifetime of that. Well, that's because zinc is a cofactor to some type of enzyme that works to help us get better. So that's why they're so important. All right. So these metallic ions can activate enzymes. They're non-active if those metallic ions are not there. They're not able to do their job. Okay. So like I said, they help form the correct shape in the active site. They help bring the substrate and the active site closer together. And so they are very important. Okay. So remember, a coenzyme is a type of cofactor, and that just means that the cofactor, remember, that's the metal ions, all right, and the coenzyme is going to be an organic molecule, so it's a little bit bigger. That's often going to be things like our vitamins, okay? This is where our vitamins kind of come into play. And so, let's see. Enzymes, and this is kind of separate. You could kind of almost put this on two separate slides here. Enzymes are going to almost always have the ending ASE as part of their name. There are a few exceptions to things that we thought were enzymes and found that they weren't, but you don't have to worry about those. They generally name the substrate acted on, or they're named for the reaction that they catalyze. So that's very handy. They kind of tell you what they do, and they do different things like transferring hydrogen atoms or electrons, carbon dioxides, amino groups. Many of them are derived from vitamins, and they're classified and named according to characteristics like their site of action, type of action, and substrate, etc. Prefix stem words are going to be derived from that certain characteristic, like the substrate acted on. Okay, so like sucrase works on sucrose. All right, really, really handy there. It's one of the few things in science that actually makes great common sense. Yeah, it is what it is. Okay, so let's put what we've talked about to use with a little prior knowledge. Which of the following is considered to be an anabolic reaction? Select all that apply. Krebs cycle, polypeptide synthesis, cell assembly, or glycolysis. All right, pause here and come up with your answer. All right, so let's talk about this. Krebs cycle. Krebs cycle is part of what? Aerobic respiration, where we take glucose and we break it down and we use the electrons, transfer them to something else to make energy, right? Because we're going to send them on to the electron transport chain, if you remember correctly. So Krebs cycle is going to be more of a catabolic reaction, okay? Polypeptide synthesis. What on earth is a polypeptide? I hope you know that if you took my class. Poly means many. Peptide, it generally means small protein. So this is making a large protein. So I'm making something that is assembly that's anabolic. So you should have said B. Okay, cell assembly, anabolic, all right. And then, so C as well, glycolysis. Well, what is glycolysis? Sugar for glycolysis means loosening, so I'm breaking something down. So that's a catabolic reaction. So you should have said B and C. This is looking really long, so I think I'm again going to have to split this up, but we shall see. All right, so constitutive enzymes are always present in a constant amount, regardless of the amount of substrate. But we also have regulated enzymes, and these guys are going to be produced whenever production is induced or turned on, or when production is repressed, which means turned off. And that's generally in response to the amount of substrate we have. Here's another really good NCLEX prep practice question. This is good for reviewing material, but it also helps with many of those entrance exams that you're going to have, and you can really see why this information is so important. Okay. All right. So an enzyme that catalyzes starch is called what? Whereas an enzyme that digests fat is termed what? Okay. And so if you just stop and think about these, it's going to help make sense. You can at least eliminate some. Okay. So pause. Come up with your answer. All right, so let's see, carbohydrates. So that should be coming off of a carbohydrate, which is a sugar or a starch. Lipase is going to be a fat. Maltase is going to be the enzyme that breaks down maltose, which is a simple sugar, okay? And then peptidase is that's coming from peptide, so that's a protein. So what is the correct answer here? Well, it's going to be A. An enzyme that catalyzes starch is carbohydrates for carbohydrate, so we know it's going to be A or C. And an enzyme that digests fat, well, the only thing here that is talking about fat is what? Lipase. So then it's got to be A.